And now we go to functional connectivity. Okay, so the human brain does not look like this, but it's good to see it like this. So approximately 20% of the total energy produced by the body is consumed by the brain, even when you're not performing any cognitive task. So let's say even when you're sleeping, or you are just there bored or something like that, you still 20% of the brain, or 20% of the energy is used by the brain. So can we figure out what it is doing when we aren't doing anything that we feel important, and how? So let's find out. So let me introduce to you resting state fMRI, which is what Dr. Biswal actually invented or discovered. So there was this field of fMRI, which was task based, which is the subject used to perform a task inside the scanner, and then the brain signals corresponding to that were taken and analyzed. But Dr. Biswal first found out that we can also use these fMRI signals when the subject is not performing any task, or in other sense, we don't know what the task is. He is maybe thinking about food, thinking about McDonald's, thinking about, I don't know, Christopher Nolan, but we don't know. And uh, still is fMRI I mean, still the signal that is coming out, can we analyze them? Can we make any sense of the brain signals using these what is called as resting state images? So the person is lying in the scanner. He may be a patient, he may be a subject, maybe a healthy control, maybe anyone. He's just lying inside the scanner, and what the scanner gives you is this 4D image, the three spatial brain dimensions across time. So it's like a movie, brain movie, and that's why it's 4D. So now you can visualize this as follows. So you can visualize this as one 3D object with a lot of voxels, and every voxel having a time series. So I've just converted this 4-dimensional representation into, in some sense, pseudo 3 dimension, with every voxel having the 4th dimension, which is the time series. All okay with this? So basically over here, every box has a time series, or is a vector now of time series. Cool. Now, what did Dr. Biswal find out? So this is his most cited paper. So what he found out was, let's say we take a voxel in the brain. Let's say it's this B voxel. Okay, I'm showing you just one slice. Okay, it's an axial slice of the brain, and let's say I take a voxel over here, and the time series corresponding to that voxel looks like this. What I do is I correlate this time series with every time series that is there, with every voxel time series in the brain, and I show you the correlation over here. So every red is a correlation that is greater than 0.35. So these voxels, let me show you. So these voxels are having time series that are having correlations greater than 0.35. You can see they are similar. So when this time series is high, this is also high. When this is kind of low, this is also low, and so on. So it's not exactly the same, it's similar. So that's what we say as correlated. And then there are certain voxels which are anti-correlated. That means when this increases, this is decreasing. But the idea is when this goes up, this goes down, and so on. So there are anti-correlated voxels and correlated voxels. So and then there are voxels which are not correlated, which are like within minus 0.35 to plus 0.35. So when you look at the map of all the voxels that are correlated to the seed voxel, that is the B seed voxel, you get this connectivity, you get this map which are at spatially different locations. So B is so very far from A or this particular region, but still they are having the same time series, the similar time series. So this is the idea of functional connectivity, that there's no physical connection between the two, or there's no known physical connection between the two, but somehow they are acting similarly. So if one particular neuron fires over here, the other neuron at a distant location is also firing up. Again, fMRI does not actually record neuronal signal, or it does not actually record the electrical firing of neurons, but it is an indirect measure. So if neurons require if neurons fire, they require oxygen. So the local region around that voxel has, or the brain supplies more oxygen to that particular region because neurons will require it, and we measure that blood oxygen signal. So the idea is wherever a neuron fires, the blood oxygen over there is high, and that's what MRIs can detect, or fMRIs can detect, and that's what is plotted over here, the blood oxygen level signal. Anyways, the important point over here is that these signals are similar but at a different location, and it turns out this is what is the motor network. So let's say if you were in a scanner doing a finger tapping task, if you are tapping your fingers, it turns out that the same network is there when as like this. So this is what is called as motor network. To give a little more details, what people do is, subjects are in the MRI scanner, that they have the experimenter, let's say I have this paradigm where I say, okay, first 10 seconds don't do anything, the next 10 seconds tap your finger, the next 10 seconds don't do anything, tap your finger, don't do anything, and so on. So I have this paradigm, don't do anything, tap your finger, don't do anything, tap your finger, and so on. And given this time series, which is my experimental paradigm, I correlate this time series to all the voxel time series that I have in the brain and get a similar map which is correlated to that experiment paradigm time series. And it turns out that if you do that, I mean, if you map the motor of function finger tapping task fMRI to this, it's similar. So that's how Dr. Biswal first discovered motor network in the resting state signal when the subject wasn't doing anything in the scanner. Cool. Okay, so yeah, so that's about functional connectivity. Now, it's not just the motor network, you could have different networks. So there's something called as the default mode network, which is quite famous. So the default mode network is thought to be the network which is active when you are mind wandering or you are bored. Basically, when you're not thinking about something in particular, it's when you have these chain of thoughts. You think about food, and then you think about something else that reminds you of pizza, that reminds you about some person you met at pizza place, something like that, and that's the trail of thoughts. So when you are just thinking or mind wandering, your default mode gets activated. This is the visual network. This is the frontoparietal network, which is involved in decision making, goal oriented tasks, and all of that, and the motor cortex. Oh yeah, over here the color scheme is different. So over here, the more the yellow, the higher the correlation, and the crosshair, the cross point shows you the seed voxel. So this particular voxel is correlated to every voxel in the brain, and you get this map. Okay, so but this is I mean, what if I want to analyze all the networks at once? So by doing this, I just analyze one network at once. So default mode, it looks like this. Visual network looks like this. But I want to like combine all of this and work with all the networks at once. So what should I do? So this is like one to many, one seed voxel connected to many, but I want many to many. I want every voxel connected to every voxel, which is like the entire brain functional connectivity. So that's what we desire. But the problem is that there are a lot of voxels in the brain. There are about 100,000 voxels, depending upon what is the dimension or what is the resolution that you are working with. 1 millimeter MNI space, 2 millimeter MNI space, and so on. So there could be 100,000 voxels. If you calculate the correlation of every voxel with every other voxel, that means you will get a 100,000 cross 100,000 matrix, which is one voxel correlated to all the other 10,000 voxels, that's one. The other voxel correlated to every 10,000 voxel, that's two. And like this, you have a 10,000 cross 10,000 matrix, which is huge, which is really huge. And my laptop sometimes cannot even handle small things. I don't know how it will handle 100,000 cross 100,000 voxels. So what we can do is use a brain atlas. So a brain atlas is predefined by other neuroscientists in the field, which give you regions that are similar. So here every color corresponds to one region in the brain that is computed using some criteria. Maybe they have similar time series always. So actually you can see, so if the spatial location is very small, the time series are mostly correlated. So why waste space with all these highly correlated time series? Let's just put this into a region, and that region is what we are going to deal with then. So this can reduce our dimensionality to a great extent. So there are a couple of brain atlases that are proposed. I know of at least 11 different brain atlases. Depending upon the different criteria, they have created these different atlases, which are of course for different applications. Now let's say we fix one particular brain atlas, let's say this brain atlas. What we can now do is we can take the average of all the voxels that constitute a particular region and then work with the average time series. So now we are reducing our dimension from, let's say, 100,000 voxel time series to some n region time series. Now that's easy. So if there are n, let's say if there are n regions, I can compute a correlation matrix of n cross n, where n is in the range 100 to 1000, which is something that my computer can handle. And this is how it looks like. Now if you actually see here, there are some regions which are highly correlated. You can see a box over here, you can see some small boxes over here, some small boxes over there. It turns out these are the different networks that I was just talking about. So this over here is the visual network. That means all the regions that are over here, the regions corresponding to these time series, belong to the visual network, or they belong in the visual region, the backside of the brain, and so on. So the default mode over here, the red one, corresponds to the default mode regions that are there across brain. So now instead of working with the 100,000 cross 100,000 dimension, we are working with the n cross n dimension, where n is a small number between 100 to 1000. another thing is, now this is a symmetric matrix because correlation. So over here, let's say if I pick out one particular element over here, it tells me the correlation of, let's say if this is the 10th region, so it tells me the correlation of the 10th region with the 5th region, let's say. Okay, so 10 comma 5 is nothing but the correlation of 10th region time series with the correlation of 5th region time series. So correlation of 10 comma 5 is the same as correlation of 5 comma 10 because correlation has nothing to do with the position if you put 10 first or 5 first. So because of that, you get a symmetric matrix. So the elements that are there above the diagonal are exactly the same as the elements that are there below the diagonal. So it's again waste of storage. Why do I keep double the data? So I can just remove the lower triangular part and use the upper triangular only, and I can just use the lower triangular. You can take any of these and then make it into a vector. So basically putting the first element here, this particular row, this particular row of element here, the next row below it, the next row below it, and so on. I get a vector over here, and that is a representation of one fMRI scan. So this is known as the functional connectivity vector of the fMRI scan.